thanks for coming today for PubhD. So those of you who are new, PubhD is an event where we get PhD students and academics and other researchers to come and discuss their research with us and the general public. It's great because we're all, like, I'm from chemistry, you're from
lung cancer oncology.
Of course, I know that. Lung cancer. There's people here from English and literature and other studies, and it's great to get other universities together. PubhD was created by two guys who just wanted to hear interesting talks in exchange for pints. So that's what we do. First up, though, we have Martin Barr. He's from Trinity's Translational Oncology Department in the Institute of Molecular Medicine. He studies lung cancer, and he's going to tell us about it.
Thanks.
Thanks, Barton.
Good evening, everyone. My name is Martin, and I'm a clinical scientist in the Lung Cancer Research Group in the TTMI on the hospital campus here in town in St James's Hospital. So I'm just going to give you a bit of as much of a lay term as possible, just a bit of an idea of what goes on in our group and what kind of research we're doing in lung cancer. So I'll just start off by giving, for those who aren't really familiar with lung cancer and its incidence here in Ireland, but it's the biggest killer worldwide and in Ireland. It's the most common cause of cancer, all cancer-related deaths globally and in Ireland, and it accounts for more deaths every year than prostate, breast, and colorectal cancer here in Ireland. And as of 2011, it overtook breast cancer as the most common, as the biggest killer of women here in Ireland. So, as a lot of you know, there's various risk factors that cause lung cancer that are known to date, and I suppose the biggest one of these is exposure to secondhand smoke, which we're all aware of. And thankfully, the tobacco control program has now, implemented this in the last few years. And as a result of this, in the recent report, they have shown that by inhibiting the smoking, the ban of smoking in public places, this has decreased in the incidence of lung cancers to a certain extent. Now, following on from secondhand smoke, you also have exposure to radon gas. You've got environmental pollutants, and also you've got people who may be predisposed to lung cancer if they have a strong genetic pool within their families. Now, unfortunately, even though lung cancer is the biggest cancer killer worldwide, there's still a lot of stigma associated with lung cancer, and this is due, or by association, with tobacco use. It's underfunded, even though it's the biggest cancer killer worldwide. People don't want to know about lung cancer. People don't want to fund lung cancer because once they hear lung cancer, they say, "Oh, he deserves it. He smoked all his life, so why should I fund him when all he has to do is stop smoking?" And again, it doesn't receive as high profile as other cancer types such as breast cancer and other types of cancers for which is quite high cure rates. So I think it's very important. First of all, from my perspective as a lung cancer researcher, I feel that there's still a lot of stigma associated with lung cancer, and although it's a choice people make to smoke, and they obviously put themselves at a higher risk of developing lung cancer because of smoking. However, there are people out there, there's about 25% of that patient population that develop lung cancer that have never smoked. And again, we have to consider them as well. Basically, if you have lungs, you're likely, or you're at risk of getting lung cancer. Current treatments for lung cancer are mainly chemotherapy based, and for many years the gold standard chemotherapy for lung cancer has been platinum based drugs in combination with various other chemotherapeutic drugs. And unfortunately, a lot of patients do undergo a lot of unpleasant side effects from this toxic regimen, and there's only about 1/3 of patients that are treated with this specific type of chemotherapy that actually will have a tumour response to treatment. Now, luckily in the last maybe 10-15 years, with the advent of targeted therapies, this is a new type of therapy that's come on board and has really progressed treatment in lung cancer. So basically, down through the years, various mechanisms have been discovered within tumour cells that have shown that specific genetic mutations in lung tumour cells drive the growth of the tumour. And they have found that by targeting these specific genetic mutations, such as EGFR or ALK, which are the two main ones, there are now targeted therapies available from pharma companies that specifically target these mutations, giving patients longer and prolonged survival. And this is a great advance in progress in lung cancer research and in the clinic, However, the unfortunate thing is that these specific mutations only occur in a very, very small subgroup of lung cancer patients. Like for EGFR, for example, it's only approximately maybe up to 14% in the European population compared to Asian populations where it's as high as 38%. Whereas the other target mutation, ALK, that only occurs in about 2 to 4% of lung cancer patients. And the biggest, I suppose, as it always has been, the biggest clinical challenge is the development of drug resistance. So unfortunately, even though some patients may respond initially to chemotherapy, the tumour will respond to chemotherapy, after about 6 to 8 months of treatment, a lot of patients, the majority of lung cancer patients, will actually develop drug resistance to the chemotherapy. And there's no other options available if they don't have the other specific genetic mutations, and therefore the patients unfortunately succumb to the disease and die. So this kind of really initiated my research in kind of bringing the area of lung cancer research into a more translational level, so bringing it closer to the patient as opposed to deciphering various molecular pathways and not really having any drug targets. Because at the end of the day, I feel like it's great, you're adding knowledge to molecular pathways maybe that are involved in the development of tumour growth in lung cancer patients, etc. But unless you have something to target, you're not really going to bring that forward anywhere and contribute to any kind of developments in new drug therapies. So this became a very big interest of mine in terms of research. So I initiated a program of chemotherapy resistance in St James's Hospital. In order for me to start out doing this, I asked myself, well, what do I need? I need some kind of a model to work on initially to find out what's causing these mechanisms of resistance and what can we discover in terms of targets that we can maybe target with a specific drug. So what I did was I set out, for any of you that are scientists, you'll know that we use in the lab a lot of the time commercially available cancer cell lines. So these are originally from a patient, they're cultured in the laboratory, and we can add drugs, etc. Long term to try and decipher various molecular pathways, etc. So what I did was I took a panel of lung cancer cell lines that were available commercially. I developed a model of cisplatin resistance. So again, cisplatin is the platinum drug that's used in patient chemotherapy. I exposed these cells to chemotherapy, i.e. cisplatin, over a prolonged period of 6 to 8 months to make them resistant relative to their sensitive counterparts. So what I had then was a panel of lung cancer cell lines representing all the specific tumour histologies of lung cancer. I had the cisplatin resistant lung cancer cell line together with its corresponding sensitive cell line. So this then would act as a valuable tool for which I could study the various molecular pathways. And another area I was interested in was cancer stem cells, because for many years it's known that cancer stem cells within a tumour is what causes them to become resistant. So this was a great advantage to myself and to others in the department, because we now had an in vitro model system where we could mimic what was happening within the lung tumour in the laboratory setting. And one of the first translational studies I carried out was looking at microRNAs and seeing whether or not microRNAs could perhaps predict what patients would respond to chemotherapy. So microRNAs are very, very small RNA species that act by, they target multiple genes, which in turn can regulate a number of different cellular processes involved in cancer. So what I done was I profiled using specific profiling arrays, where this consisted of over 2,000 microRNAs, and these microRNAs were very novel kind of RNA species, and there's been a lot of focus and research with these at the moment, in particular in relation to drug resistance. And what I got from that was, out of 2,000 microRNAs, I narrowed this down to 5 specific microRNAs. So basically I had a signature, a microRNA signature, which we found these specific microRNAs to be significantly regulated, either highly expressed or very lowly expressed in the resistant cells compared to their sensitive cells. So what I was able to do was take this 5 microRNA signature, find out which microRNAs were up and down. For example, in the cells that were resistant, if we had microRNAs that were upregulated, we were able then to add inhibitors to these microRNAs and see what were the effects on the tumour cells. And what we found was that where we had increased expression of these microRNAs, I was able to inhibit them in the resistant cell lines. And what happened was, when I added back cisplatin chemotherapy to those specific cell lines, I was able to overcome cisplatin resistance and increase quite significantly the cell kill. And likewise for microRNAs that were downregulated in the resistant cell lines, I was able then to add into the cells synthetic microRNAs of the same type, so therefore overexpressing the microRNAs. And again, this helped overcome the resistance phenotype, and we were able to kill the majority of the tumour cells. So this was very promising in terms of these microRNA profiles. So what we're trying to do at the moment is we have already designed a study to bring into the patient setting. So what I want to do next is, in collaboration with the medical oncology department in the hospital, is to recruit lung cancer patients who are in advanced stage disease. When they come into the clinic, take their blood, profile these 5 specific microRNAs in the blood, and then after every cycle of chemotherapy, so basically patients receive chemotherapy in cycles, so maybe 1 every 4 weeks. So we'll take blood from the patient as they go through the various cycles of chemotherapy until they progress, and look to see how these microRNAs differ in their expression over time, with the ultimate aim of seeing, okay, we now know that, you know, as patients develop resistance to cisplatin chemotherapy, these specific microRNAs are changing, they're either going up or they're going down. So the idea would be that prior to receiving chemotherapy, if you had a companion diagnostic test that we could use on the patient using a blood sample, we could profile these 5 microRNAs, and if the profile was similar to what we have found in our in vitro model, cell model of increased expression of these microRNAs, we would then know that this is predicting that this patient will not respond to cisplatin-based chemotherapy, and therefore prevent having that patient have to go through multiple rounds of chemotherapy, not have any effect on the tumour, but just making the patient very sick with a very poor quality of life. So that now is at the stage where we're looking for funding to bring this forward into the clinical setting where it has some kind of real meaning to lung cancer patients. The second kind of big area that I've been working on as well is, as I said, cancer stem cells. So for many years there's been a lot of focus on cancer stem cells in solid tumours. And these are basically, like in your normal tissues, you have normal stem cells, and these are involved in the maintenance and repair of tissues within your body. However, in tumours, cancer stem cells are a rare population of cells that populate the tumour, and they have the unique ability of being able to self-renew, differentiate. They can also initiate and maintain tumour growth. So what happens is when patients become resistant to radiotherapy or chemotherapy, it tends to be down to these specific population of cancer stem cells within the tumour population that just don't die as a result of the treatment, and they continue to grow and multiply, and this is why then the tumour is no longer responding to the therapy, because it's just multiplying and not getting any smaller. So what we did was we set out to look at a number of different cancer stem cell markers that had already been discovered down through the years and previously published, to see what their expression was in our resistant cell line model, to see could we actually find one specific cancer stem cell marker that we could then target as a means of getting rid of that cancer stem cell population within the lung tumour. And after a lot of experimental analysis and many different experiments, there was only very modest increases in the kind of the standard cancer cell markers that are published out there for solid tumours. However, interestingly, we did find one specific marker called aldehyde dehydrogenase, or ALDH1, and this was significantly increased in our cisplatin resistant cell lines compared to the sensitive cell lines. So right away, this was to us something that we would want to look further into, and after different types of cellular and molecular functional assays, we found that in tumour cells that had high expression of ALDH1, these were the ones that were driving the whole drug resistance within the tumour. And then it was just very interesting because in the middle of all of this, we had come across a paper that was looking at a drug called disulfiram, and this is used in the treatment of alcohol addiction. And what it does, its pharmacological action is actually to inhibit aldehyde dehydrogenase. So we thought right away, this is perfect. This is a target. This is a drug that targets what we're looking at, ALDH1. It's been used for 60 years in the treatment of alcohol addiction, so therefore it's been FDA approved. All its tolerability, toxicities, etc., these are all known. So it's basically, we thought this is a way of potentially repurposing a drug that already exists for another disease, i.e. alcohol addiction, and apply this to cisplatin-resistant lung cancer. So taking this on board, we went ahead, we used disulfiram in combination with cisplatin chemotherapy on our resistant model, and to our surprise, this had huge effects in the tumour and killed a significant amount of the lung tumour compared to the drug on its own or chemotherapy on its own. So again, because it's already FDA approved, like we know its pharmacokinetics, we know it can be given orally, so therefore it's very convenient to administer to patients should it go into clinical use. And more importantly, unlike many chemotherapeutic agents, disulfiram has the ability to cross the blood-brain barrier. So this means then that it may potentially be active against brain tumours. And unfortunately, a patient with lung cancer, if the cancer is going to spread to other sites in the body, one of the main sites it spreads to as a secondary cancer is to the brain. So again, this fitted really nicely in with what we were looking at. So we published this study just last month in Oncotarget, which is a scientific journal, and we're now in the process of trying to bring this forward into an in vivo model with the potential of perhaps initiating a phase 1 or perhaps maybe phase 2 clinical trial in the near future. Because it is already FDA approved, we're now repurposing this drug that was once used or currently used for alcohol addiction into the setting of cisplatin or chemotherapy resistant lung cancer. So we're very excited about this at the moment, and we hope that maybe this actually does continue into the future and lead into a clinical trial. So it's nice to see that whole kind of lab-based discovery moving forward then potentially into the clinical setting. So of course all these translational studies wouldn't be possible without the use of human samples, and we're very fortunate in our lung cancer research group in St James's Hospital in that we developed or established a lung cancer biobank back in 2006. So what this basically is, it's basically lung cancer patients are coming into the clinic who are amenable to curative surgery to remove their lung tumours. We get informed consent for the patient to give us permission to take a piece of their normal lung tissue or their tumour tissue, their normal lung tissue, together with pre-surgery bloods from which we isolate plasma, serum and white blood cells. So what we were able to do, we were able then to validate a lot of the markers of resistance that we found at the bench in the laboratory, validate these in vivo in the patient setting, basically using their lung tumours to see, okay, in the real lung tumour, are we getting the same levels of expression? So basically we're getting very high expression in the tumour, no expression in the normal lung tissue. Well, this makes it a very good target then for a specific drug. So that has really helped us along the way to make this work more translational and to kind of bring it closer to the patient setting. And again, we feel it's very important then that patients are aware of what biobanking is, and if you're a patient unfortunate enough to be diagnosed with lung cancer coming in for surgery, that if you feel that you would like to give something of your tumour to research that may help in the future come up with novel diagnostics or novel treatments, that the biobank is something they can talk to us about if they want to consent for this. So as a result of this, this led us to generate or establishing a lung cancer charity called Target Lung Cancer, or TLC, which we set up 2 years ago, where our mission really is to educate the public, not only educate them on lung cancer and the biobank and what they can do and how they can participate as a patient to kind of help drive new discoveries forward, but also to fund young researchers and to kind of train those to international standards, and again to improve patient care for lung cancer patients who are coming into the oncology clinic in St James's Hospital. So that's just a bit of an overview of some of the work I'm doing. So sorry if I made it a bit too scientific or whatever, but any questions feel free to ask anyway.
An A, or some person can have a T. So you have an A allele or you have a T allele. And so the different frequencies of alleles differ from different populations. And that's basically the basics of population genetics. Mutations differ in their frequencies between different populations. And there are a number of different reasons why this can happen. So there are 4 main different reasons, all stolen off of Wikipedia. The first one is mutation, which I've already covered. The second one is selection. So some populations have different genetic mutations that are at different frequencies because one of those variants can help them survive in an environment that they find themselves in. So classic example are populations in the Himalayas, because they've had to adapt to the high altitude environment that lives there, that they live there. Anyway, That's selection. So the third one is admixture, which simply just describes the mixing of two different populations. So this is what's happened in America, where populations from across the world have mixed into America. So America is quite admixed. And then the fourth and final one is genetic drift, which is a rather nebulous term, but the best way to describe it is through an analogy that involves a bag of balls. So, if you imagine a population as a bag of balls, and on all of these balls have different colours, and if the different colours represent the different types of alleles that you can have at a different site. So these balls can be blue, they can be yellow, they can be green, they can be purple, they can even be blue. No, red. I crossed off, yeah. There can be lots of different colours, which is the point of the analogy. So every generation, as part of creating the next generation, you take a sample of the genetic variety in that gene pool, or that bag of balls, and by chance, and by random chance, some of colours are going to be oversampled than others. And so by chance, next generation you can oversample red balls, for example. So red balls become more frequent in the next generation. And this is an entirely random process, but it relies on the last generation. So you can imagine that each generation, the drift of allele frequencies can change and can either go up or down. And this is genetic drift. Essentially, over time, populations will just accumulate different frequencies because of random assortment, and they just drift away from each other. And this is a neutral process, so it doesn't rely on any selection. It relies on a number of different things, but it's basically a neutral process. And so this is the fourth and final way that populations can differ, and they just kind of move apart over time. And so those are the four different ways that populations can differ, and we understand now what mutations are. At least we know what single nucleotide polymorphisms are. So how do we study this? So there are two broad methods that we can use to investigate the genetic identities of an individual. And this process is called genotyping. So this is the investigation to see what class, what type of mutation you have at a particular point. So you can either sequence everything in the genome, which is called whole genome sequencing. There's exome sequencing, but we're going to ignore it for the point of this conversation. Whole genome sequences is everything, so it's rather exhaustive, but you get all the data. What you could do for a cheaper method, and which is also a little bit more heuristic, you can take a subset of the SNPs that are in the genome, and these SNPs will represent the genetic diversity across the genome, or at least the common diversity in the genome that's covered across all of most of the human population. And so this is called microarrays, and this is what the majority of population genetics is based around. So for example, the common microarray genotypes an individual by 700,000 different points in the genome. So each individual has 700,000 data points, and you might have a cohort that has maybe 1,000, 2,000 individuals. So now you have a data set which has 2,000 individuals, each of one that can be described in 700,000 different ways, which is a rather complicated and very high data data situation, which is why population genetics tends to use quite high computational programs, and is usually placed on a supercomputer instead of doing it on the laptop, just because there's just so much data to go through. So now we know what mutations are, now we know how to do how populations can differ and the processes that can make them differ, and now we know how to investigate them. So now I can actually start talking about my PhD. So my PhD is based on investigating the population genetics of Ireland, i.e. the population structure. So population structure literally just describes how populations are differed. So if you have a group of organisms that haven't genetic structure, they're basically just everyone in that population has roughly the same allele frequencies. But if you have different groups within that group of organisms that have different allele frequencies, those are the populations. So that population can be known as structured. So what I'm doing is trying to investigate whether Ireland is structured or whether it's just one homologous mass of individuals, one population. Spoiler alert, it's not. So we took a load of individuals and in a cohort called the Irish DNA Atlas. And so what we didn't do is just take random Irish people from the street and take their DNA. One, that's unethical, and two, it's really bad study design. I'll let you decide which one's worse. So what we actually did was recruit individuals that have all 8 of their great-grandparents born within 50 kilometres of each other, which means that their genetic ancestry and their genetic variation that's captured in that one individual is representative of specific regions in Ireland. So what you then can do is not just compare Ireland to just neighbouring populations such as Britain or continental Europe. What you can do is compare different regions in Ireland to each other, and then you can start teasing apart how different genetically different regions in Ireland are. So that's what we tried to do, well, that's what we did in our first kind of round of analyses. What we did was compare these Irish individuals to British individuals, and we run this clustering analysis. So what it did was take this combined data set and then try to fit everyone in their own little population. So trying to fit people that are more genetically alike together than they are genetically distant from everyone else. So these are kind of small subpopulations within the population of the British Isles and Ireland. And what we found is that the clusters that we observe within Ireland are geographically stratified, i.e. that they're found in geographic regions and they're not just spread randomly across Ireland. So we have clusters that describe the boundaries of Munster near perfectly, which is quite cool. We have clusters that describe the old kingdom of Leinster, of Ulster, and also of Connacht, and also around Dublin. So this is showing us that sociological boundaries in Ireland are the main determinants of population structure within Ireland. They're the main determinants of what make people in different regions of Ireland different from each other. So these genetic differences are really slight. We have to use really powerful statistical techniques to be able to detect them, but we can detect them. It's quite cool. But what we also found is that a subset of our Irish individuals looked like they were roughly half Irish and half British. And they were also found mostly in the north of Ireland. So you can imagine that they might be due to an admixture effect. So we tried to use, I keep on using try, we did. You succeeded, Ed. So we ran this analysis detecting admixture, and we found and we dated this admixture event to be sometime in the 17th or 18th centuries, which you might know coincides with the Ulster plantations in the north of Ireland. So we seem to have found the evidence of the genomic impact of the Ulster plantations on the genomic landscape of Ireland. But then we went a step further and compared Ireland to different parts in Europe. We found that the closest, genetically speaking, group within Europe is French individuals from the northwestern tip of France in Brittany, which is classically known as the last Celtic isolate found within France, which Ireland is a Celtic population, and Brittany is known to be genetically close to other Celtic populations within British Isles, so Cornwall, Wales, Scotland. So that's not too surprising. But what was surprising was a significant part of the Irish genome looks rather Norwegian, but not just any type of Norwegian. It looks like the type of Norwegian you find on the west coast of Norway, which is where the Norse Vikings came from. So we decided to see whether we could detect that admixture, see if we can date that time of admixture, and the date of the admixture arrives just around the time, just the end of the first millennium, which is when the Norse Vikings were appearing around the Irish coast. And this Norwegian ancestry is spread throughout Ireland, which means that there has to be a movement of people post-dating the Viking invasion of Ireland to spread that Norwegian ancestry through Ireland. So that's where we're at talking about settled Ireland. What we've also been doing, what's been alluded to, is that we've also been working on Irish travellers. So we've got a similar data set of individuals with Irish traveller ancestry, and we've been running clustering algorithms on them. So you might expect that the Irish travellers are just one population and they just all group together in one cluster, but that's not the case. So the Irish travellers, there is a small amount of surnames that are in the Irish traveller community, and certain families tend to marry within those families, but to the exclusion of other families, in a sort of clan-like structure. And we see that reflected in the genetic data. So we see, we observe 4 clusters of Irish traveller membership, and though 2 of those clusters are grouped in one group and 2 of those clusters are grouped in another group. So we'll be talking about the 2 groups mostly. So if you look at the surnames of individuals in those groups, they're the surnames that you would classically associate with marrying with towards each other, which is a nice confirmation of that genetic structure that we observe. As well as when we look at the language. So the Irish travellers speak their own language called Shelter, which is split up into 2 dialects, Cant and Gammon. And so the Cant speakers are from traditionally the west of Ireland, and the Gammon speakers are traditionally from the south-east of Ireland. And so we see that one group of the clusters exclusively speak Cant, and one group exclusively speak Gammon. And then we went a step further and tried to see what we did. We investigated whether any specific region in Ireland had a genetic affinity, or any Irish traveller group had an affinity to any specific region in Ireland. And we found that the Can-speaking group have a greater affinity to the west of Ireland, and the Gammon speakers have a greater affinity to the south or east of Ireland. So that's where we're up with the travellers as well. So kind of just to wrap things up, we've described structure within Ireland, and the structure seems to be reflective of demographic boundaries within Ireland rather than geographic boundaries, which kind of makes sense. There's no kind of like large mountain range in Ireland like the Alps that just forms a gene flow barrier. And then with the Irish travellers, we observe this kind of genetic structure that seems to be related to different ancestries around from Ireland, or at least a greater affinity to different regions of Ireland. And that's really important for genetic disease work within Irish travellers. So the community has a higher prevalence of recessive diseases, so it's quite possible that different groups of Irish travellers have different kinds of genetic mutations that cause disease. So understanding the underlying structure there will be able to, that will hopefully help the identification of specific diseases in those groups. So thank you all very much for listening. I'll be very open for questions now or after I've had a couple of pints.
Great opportunity. And I was really nervous at first because obviously there's been tons of research done on modern Halloween, so there must have been lots of research already done on Samhain, which is where Halloween comes from. Imagine my surprise and delight when there's actually very little published out there. If you manage to find some research that's been done on Samhain, usually they'll list 3 things about it. So first of all, or 4 things, we'll go with 4 things here. First of all, it's one of the 4 festivals that were celebrated in pre-Christian Ireland back in the day when everyone was farming, and you needed some sort of landmark to tell you when you needed to do something new on your farm. So whether it was planting or harvesting, moving your flocks and herds to the correct pasture. So you had these 4 festivals that divided the year for you. The other thing that you'd learn in these, whatever it is that you've managed to find with information on Samhain, is that it's when the veil between our world and the other world, the she, is at its thinnest. So it's when all of the supernatural things happen. It's when the ghosts come out, it's when the she folk come out, the fairies come out, all this scary supernatural stuff starts to happen. You've also got a time for feasting, and it's the end of the year. So the end of not just the agricultural year, but the end of the legal year as well. And most things, that's where it ends. So I was really excited because it meant that pretty much anything I did was going to be new information. So a lot of weight lifted off my shoulders. So what I decided I was actually going to do was study the myths and try and put together a landscape study of Samhain. What I wanted to do was answer a few questions. So first, by going through the myths, I wanted to know what people, what places, what were people doing at those places, and what artifacts, because at the end of the day I am an archaeologist, so I'm interested in those things that people left behind, the physical things that we can touch and study. So what artifacts can we associate with Samhain? As I went through the myths, I want to make sure I don't lie to you, so I wrote these numbers down. I read 136 myths. Of them, 24 contain Samhain. Doesn't sound like a lot, but when you consider that the other 3 myths aren't mentioned at all in the myth, or the other 3 festivals aren't mentioned at all in the myths, that number actually starts to become pretty significant. So, and now I know that there's actually some more. I've managed to find a few more, which is exciting. Of those 24, I started kind of reading through them. First of all, who are the people who are involved? Lots of mention of Cuchulainn. When I first started out this research, I was told by other researchers that all of my information was going to come from the Ulster cycle. So the Ulster cycle of myths is everything that happened in Ireland before our world and the other world, where the fairies live, were separated. So the theory was that most of my tales were going to actually come from there, from the mythological cycle. This ended up not being the case. Most of the myths were coming from the Ulster cycle. So when Queen Maeve and King Ailill and Conchobar and Cuchulainn are in their height, That's when Samhain was at its height in these myths. And people told me that I was wrong and I needed to start my research over again. So I did. And for anyone who's been told that, I don't need to tell you how exciting it is when you redo the research and it shows the same results. Pretty exciting. So Cuchulainn ends up being our big player in these myths. Next thing I wanted to know was, what were people doing? So when we think of Halloween, when we think of Samhain, we think of certain things, right? We think of, again, the supernatural. We think of ghosts. We think of witches. We think of goblins. We think of fairies. Not a whole lot of supernatural mentioned in these myths. In fact, there was almost none, which was very interesting. And basically what I came up with was 3 potential reasons why there wouldn't be much of anything mentioned. One being, maybe it just wasn't there. The supernatural element could be a later addition to Samhain. There's one possibility. Other possibility is that these myths were recorded largely by Christian monks. So they could have been looking at these supernatural elements and deciding that maybe it just didn't quite jive with their own Christian beliefs, and that it was maybe safer to just not record them. So there's the possibility that we're seeing this author bias when it was going from an oral story to an actual written literature. The third possibility is something that you see, a rule that you see in basic storytelling, even today. When you tell a story, you assume a certain level of intelligence in your audience. Whether they have it or not is another question entirely, but you assume this certain level of intelligence. So if I told you that yesterday was Halloween here in Ireland, I don't need to tell you that there were trick-or-treaters running around. You assume that there are trick-or-treaters running around. So there's the possibility that the supernatural is not mentioned because the storyteller is assuming that the listeners already know that it's happening, that it's just part and parcel of Samhain. Unfortunately, that's not something that archaeology can prove. So there's that. There's what were people doing? Moral of the story there. People were actually getting together and having festivals. They were having feasts. You had to go to the home of your king. If you didn't, we have 2 separate myths that say you would go crazy and die within 3 days if you didn't go to the feast held by your king. No pressure, guys. Go to the feast of your king. The other side of that was you had to go specifically with your female equivalent. So, men, you have to find that woman who's she's not just a lady, she is your social and economic equivalent, your equal. And she had to go with you to this festival or you're not allowed in. And as we just said, you'd go crazy and die. So, no pressure, once again, find your lady. So lots of feasting, lots of festivals. That's what people were doing. They were also taking some journeys at this time. It was a good time to take off. Your farms didn't need a whole lot of attention at this point. So then we also wanted to know what artifacts, not a whole lot unfortunately, not much that would survive archaeologically anyway. And finally, I wanted to know what places, because that's where my archaeology is coming in. I'm doing a landscape study of Samhain. So I want to know, where are these places? I want to map these places. And I had some unexpected results. And don't worry, I'll pass this around so you guys can all see it if you really want to. But for those of you who are close enough to see, I ended up with some really weird clustering that just really it brought up more questions than answers. And we can pass that around. Part of the problem was that I didn't have a good definition of what is a Samhain site. Basically anything that could be just vaguely associated with Samhain, I was throwing on the map. Another issue is we don't know where all these places are. Even today, we don't know where they are. Some of them may have never existed in the real world. They may have been part of the She. And that did happen. We do know a few places in the myths that were actually part of the She, not part of the real world. So it becomes very difficult then to try and create a map of these places. So my master's thesis really created a lot more questions than answers, which was a great thing because it meant I had something to do for a PhD. So what I did was I moved over here to Ireland, obviously, and I decided that what I wanted to do was revisit these myths. And so that is actually in my future plans, is to go back into these myths now that I've seen them once already and kind of have a basic idea of what to expect, and sort of revisit them, and this time with a better definition of what exactly is a Samhain site. After that, I'm also currently going through the more modern folklore. So I didn't visit the folklore at all for the master's degree. But here in Ireland, you had this amazing thing. It was the Irish Folklore Commission. It was in operation from the 1930s through the 1970s. And what they did was they sent people out into the field, folklorists, anthropologists, and volunteers, anybody they could get, and they sent them out into the field to collect folklore. So this could be stories, it could be traditional medicine, it could be recipes, anything that dealt with the day-to-day life of the common people. And they collected all this stuff. It's currently housed at UCD in the folklore department. Literally thousands of documents, and there are hundreds of pages on Halloween. Most of it has never been touched, not since it arrived in the folklore department. So, venturing into that, with the folklore, what we can help to do as far as this landscape study goes and trying to clear up that lovely map that's going around right now is two things. One, we have things like the questionnaires. In 1943, they sent out a questionnaire asking people just about Halloween in general. So what are your traditions? What do you call it? What kind of pranks do you play? What kind of games do you play? That sort of thing. And from those, we can actually get concentrations of places where you see traditions. So it's not going to be a specific location, it's going to be a concentration area. Then from the more traditional folklore, from the stories that people were telling, from the school's collection where little school children actually went out and asked people about these things as well, from that we can get the specific locations. That mountain over there where the witch comes out every Halloween and we're all scared of it, that goes on the map. The hope then is to be able to look at all of these sites and compare them to see, first of all, is there an archaeological site associated with it? Something there physically on the landscape that would keep people interested in Halloween, that would keep those traditions going, particularly before you start seeing these traditions kind of coming over from the United States, before you start seeing all that. So perfect there for the folklore department. So is there some sort of something physically on the landscape that's keeping that alive? Hopefully an archaeological site of some sort. So then is there some sort of some sort of similarity between these sites? So for example, are there passage graves on a lot of them? Finally, I want to know, is there death associated with them? Because at the end of the day, Halloween is a festival of the dead. Samhain also appears to have been a festival of the dead. And many of the sites that we do know, such as Tara and Rathcrohan and Croghan Hill and Hill of Ward, all have associations with death. They all have burials on them, which may or may not be significant, and hopefully we'll see it carry through into the actual map that I'm hoping will happen, and hopefully be a little clearer than what happened there with the master's degree. All right. I believe that's my 10 minutes.